Right, you're ready to go.
I'll just get the PowerPoint here. And can everyone see that?
Yeah, I can see it. Yeah, that's perfect. Yeah.
Great, thank you very much. Good afternoon, ladies and gentlemen. My name is Brian Maloney. I'm a fourth-year dental science student from Trinity College, and I'll be speaking this afternoon about my project, the potential of nicotinamide N-methyltransferase as an anti-cancer target in cultured human oral squamous cell carcinoma. Oral cancer is the sixth most common cancer across the world, with a 5-year prognosis of about 50% in advanced cases and treatment options limited to surgical excision, radiotherapy and chemotherapy. A more targeted approach may prove a useful adjunct to already established cancer therapeutics. One of the hallmarks of cancer is altered metabolism. Despite the upregulation of glycolysis that is seen in cancer cells, termed the Warburg effect, the oxidative phosphorylation pathway has been shown to be fundamental in tumorigenesis. Thus, this particular metabolic pathway may prove a potential therapeutic target in the treatment of oral squamous cell carcinoma if a target within the pathway can first be identified. And one such target we've looked at is nicotinamide N-methyltransferase, or NNMT for short. This is a cytosolic enzyme shown to be upregulated or overexpressed in a variety of human cancers, and has been recently investigated as a potential anti-cancer target by several laboratories. This enzyme has been shown to be involved in the biosynthesis of NAD+, thus altering protein methylation profiles and promoting oncogenesis. Notably, its dysregulation has been shown to affect the proliferative, migratory, invasive, and differentiation potential of various cancer types. Given its well-documented role in tumor progression, clinical outcome, and drug sensitivity, NNMT represents an attractive drug target for several neoplasms that are characterized by heightened NNMT activity. A recent in silico analysis of the crystalline structure of this enzyme by our collaborators in the Technical University of Dublin has identified several selective small molecule modulators of this enzyme with drug-like properties, and the effect of 2 of these inhibitors on the viability and metabolism of an oral squamous cell carcinoma line were investigated as part of this project, a novel task which has not been done before. So the primary aims and objectives of this project were first to determine whether this enzyme is expressed in 2 distinct cell lines. The first, an oral squamous cell carcinoma line, or SCC4 cells, and secondly, a pre-malignant dysplastic cell line termed DOC cells. Following on for this, we wanted to ascertain whether the inhibitors that we chose had any effect at killing these above cells, and this would then allow us to calculate an IC50 for either compound. And finally, we wanted to determine the effect these inhibitors might have on cellular metabolism, namely respiration and extracellular acidification rate, which is an indicator of glycolysis in the aforementioned cells. In order to carry out the project, both cell lines were cultured according to standard protocols. The expression of NNMT in both cell lines was ascertained using the Western blot analysis. Viability was determined with the Almer Blue assay, and respiration and extracellular acidification rate were determined with the CRS-XF analyzer. As mentioned, the inhibitors that we chose were identified as a result of a pharmacophore analysis of the active site of NNMT, and we refer to them as inhibitor 1 and inhibitor 4 here. The other compound we used was 5-amino-1-methylquinoline. This was identified in a previous study by Neil Canton et al. And used as a proxy negative control in this project. So the first of the results comes from the Western blot, or the immunoblot analysis. This allowed us to see that the enzyme nicotinamide N-methyltransferase is expressed in both the dysplastic DOC cells and the malignant squamous cell carcinoma cells. This can be seen by the dark black bands represented below. A positive and negative control are also used for comparison, and these come from breast cancer cells. Once we knew that the enzyme NNMT is expressed in the cell lines, we wanted to determine whether inhibitor 1 or 4 had any effect at killing the cells, and this was done with the Almer Blue assay. The cells were treated with a concentration range of 0.07 micromolar up to 10 micromolar for 24 hours. And the collated data below for both inhibitors and both cell lines show that there was no significant cytotoxic effect from either inhibitor on either of the cell lines, and this precluded us from calculating an IC50 for these inhibitors. And the final part of the project looked at the effect of these inhibitors at a bioenergetic level. This is done using the CRS-XF analyzer. This quantifies ATP, or energy production, through 2 pathways. Firstly, the mitochondrial pathway, as shown below. This is determined using the oxygen consumption rate. On the lower left, we can see the oxygen consumption rate before and after treatment of these cells with the inhibitors in the squamous cell carcinoma line. The blue cells were the control cells which received no inhibitor. Those in black received the proxy negative control, and the cells in red and green received inhibitor 1 and inhibitor 4 respectively. Following the injection of 10 micromolar shown by the first demarcation in the graph, there was a significant reduction in the oxygen consumption rate in the cells that received our inhibitors with no change in controls. And similarly, following the injection of 20 micromolar, we see another significant reduction. Finally, rotenone antimycin A is injected. This shuts down the electron transport chain and gives an indication of non-mitochondrial energy production. And on the right-hand side, we see similar results for the DOC cells, a significant reduction in oxygen consumption rate at 10 and 20 micromolar, and then all cells approximate one another following the injection of the rotenone antimycin A. And the final parameter measured was the extracellular acidification rate, which is an indirect measure of glycolytic flux. On the left-hand side, we see an increase in the extracellular acidification rate following treatment with our inhibitors. And on the right, for the DOC cells, similarly, we see an increase. Thus, in summary, the results of this study have shown that the enzyme NNMT is expressed in both an oral squamous cell carcinoma line and a dysplastic cell line. Neither inhibitor 1 nor 4 had a cytotoxic effect at the concentration range and incubation times used. The inhibition of this enzyme by the selected small molecule modulators was shown to attenuate cellular respiration while concurrently increasing the extracellular acidification rate, thus interfering with energy metabolism. Therefore, in summary, the metabolic data that we've gathered suggests that the inhibition of nicotinamide and methyltransferase may prove a potential therapeutic target in the long-term treatment of oral squamous cell carcinoma in combination with the conventional therapies. However, this was a proof of concept and more research is evidently needed. This would involve looking at the expression of this enzyme in the healthy primary gingival keratinocytes. This would give a baseline control for comparison with the expression in the other cell lines. More work could be done on the inhibitors themselves, looking at higher concentrations and incubation times and see if we can calculate an IC50. An EC50 of the inhibitors could be directly measured on the isolated enzyme. And then finally, the importance of this enzyme in oral cancer could be investigated. This would involve looking at knocking out this enzyme in the cancer cells and subsequently assessing for its absence of cancer phenotype. These are some references, and I would just like to thank the PI on this project was Professor Richard Porter in Trinity College Dublin, Marlena Gravaraki and Mary Rooney, again, supervisors and postdoctorate students in the lab at the time. And I'd also like to thank a number of members of the Dublin Dental Hospital for the funding, as well as helping with carrying out the project, Ailish Lapp, Claire Healy, Michael Sullivan, and Professor Brian O'Connell. Thank you.
Thank you, Brian. I'm just going to stop the recording here now.